Duchenne muscular dystrophy and Huntington chorea are two hereditary diseases where one of them is observed in a family. Document 1 summarizes the mode of transmission of these two diseases, and Document 2 represents the genealogical tree of this family. This is Document 2. For Document 1, show mode of transformation of this disease. Show if this disease is allele by a recessive allele or dominant allele. Duchenne muscular dystrophy, a disease controlled by a recessive allele carried by the sex chromosome X. And Huntington chorea, a disease controlled by a dominant allele carried by an autism. Show, referring to Document 2, that the disease affecting this family is dominant. So it is Huntington chorea, a disease controlled by a dominant allele carried by an autism. So, for Document 2, if this family, the disease of this family is recessive allele carried by the sex chromosome X, so for sure, father to this family, this allele is X affected, and Y, and the mother of this family, this allele, her allele is X affected, and because recessive allele, so for sure the second X is affected. For the kids, healthy female or healthy sister is affected, so is transformation X affected, and O X other X affected, so X affected from her father, and second X is affected from her mother. 2 is X affected from his mother, and for sure Y from his father. Or 2 is healthy female. So it's not the case, because healthy will be shown in dominant X, which is not affected, and second X, which is affected or not affected. So it's not the case. It's not Duchenne muscular dystrophy. It is Huntington chorea. For Huntington chorea, a disease controlled by a dominant allele carried by an autism. So the father of this family will be capital A, which is dominant, and short a, and for sure mother of this family, capital A and short a, dominant, which is dominant allele carried by an autism, and short a. For 2-1, transformation of dominant A from her father and dominant A for her mother, or dominant A, capital A, from her father and short a from her mother. So this allele of 2, 1 will be A, capital A, capital A, or capital A is dominant and short a is recessive. Dominant, disease, and recessive, healthy. Okay? For 2, 2, the same of the sister. A, capital A, short a, or capital A, capital A. For 2, 3, for sure is short a and short a. It is recessive healthy. So, referring to document 2, the disease affecting this family is dominant. It's here. For the answer, the two affected parent 1, 1 and 1, 2 have a healthy daughter 2, 3. This means that at least one of this parent has the healthy allele that is being masked short a by the affected one. Affected one, here capital A. Therefore, this healthy allele is recessive and allele of the disease is dominant. Okay, so the healthy allele is recessive, short a, and allele of the disease is dominant, capital A. Draw out the disease present in the family. So this family is affected by Duchenne muscular dystrophy. Which one of this disease for sure is Huntington chorea? Document 3 represents the result of electrophoresis of the DNA of the gene responsible of the disease affecting this family. So document 3, allele of the studied gene A1 and A2. Write the genotype of the fetus, so A1, A2. Specify if this fetus will be affected by the disease. It's here, okay? So DNA analyze show the presence of allele A1 and the two sister of the fetus is 2, 1, 2, 3. So 2, 1, 2, 3 has A1 and 2, 1 is affected. So the A1 is present in 2, 1 and A1 is present on 2, 3. It is healthy. Okay, so A1 because it's present in 2, 1 اللي هي affected and 2, 3 اللي هي no affected. So for sure A1 because the disease is dominant, so for sure is A1 is allele recessive healthy. Okay, and A2 present here, so A2 is the dominant allele يلي سببت الديسيز. sorry. So identify which of the allele A1 or A2 is the one responsible of the disease. So responsible of the disease is A2. Write the genotype of the brother and the two sisters of the fetus. So genotype of the brother it's 2 2, so 2 2 is affected, so A1 A2 or yes A1 A2, and the 1 1, so A1 A2 A1 A2 A1 A2 and A1 A2 because it's affected. Here A1 A1 because it's healthy, and the fetus A1 A2 is affected for sure. For exercise 2, a plasmacyte is an active differentiated B lymphocyte. It performs its role in two steps: the proteosynthesis, where plasmacytes synthesize an enormous number of the same clone of antibody, that its protein is nature, and the secretion of this antibody by exocytosis across its cytoplasmic membrane. When plasmacytes perform this step, they consume a huge amount of energy and die afterward. For question 1, pick out from the text the name of each of these two steps performed by plasmacyte. So two steps, it's photosynthesis and secretion of this antibody by exocytosis. Draw out the type of the considered specific immune response because it's present B lymphocyte and antibody, so for sure specific immune response is humoral. Indicate when structural modification that an active BL undergoes when begin differentiated into plasmacyte. So what is different into plasmacyte and B lymphocyte? Since plasmacytes it produce a lot of protein, so for sure plasmacyte has developed RER. RER is rove endoplasmic reticulum and increase in the cytoplasmic volume and disappearance of this membranous antibody and rich in mitochondria. This is a correction of question 3. In the framework of studying the mechanism of the activation of B lymphocyte into plasmacyte, many experiments are performed on 3 lots of mice. These lots are injected with different cells as shown in document 1. For question 4, describe each of these experiments. Ablation of the thymus at lot 1 and lot 2 and lot 3. For lot 1, the mice injected with antigen B lymphocyte, macrophage, and T4 lymphocyte. The result is presence of plasmacyte because all the necessary cells are present. For lot 2, the mice injected with antigen B lymphocyte, macrophage. The result is absence of plasmacyte. So what's the difference between lot 2 and lot 1? The antigen is present in 2 lots, in lot 1 and lot 2. The same for B lymphocyte and the same for macrophage. T4 lymphocyte is absent in lot 2. So because T4 lymphocyte is absent in lot 2, so the result is absence of plasmacyte. For lot 3, the mice injected with B lymphocyte, macrophage, T4 lymphocyte. So what's the difference between lot 3 and lot 1? The antigen is absent in lot 3. So because antigen is absent in lot 3, the result is absence of plasmacyte. Here is the correction oral answer for question 4. For question 5, interrupt the obtained result. When the questions say describe, you should only describe what you'd observe. Do not make any conclusion or explanation. And when questions say interrupt, you should explain the result and give a conclusion based on the observation. So for question 5, interrupt the obtained result will be interrupt the result, and the answer is here. Presence of plasmocyte are noticed only in lot 1. However, the lot 2 and lot 3 is absence the plasmocyte. Why? Because in lot 2, the T4 lymphocyte is absent, and for lot 3, the antigen is absent. So the result is absence of plasmocyte. Some mice of lot 2 are injected with interleukin 2, while others are injected with interleukin 4. These interleukins are molecules released by active T4 lymphocyte. Only the B lymphocyte of the mice injected with interleukin 4 are transformed into plasmocyte. So B lymphocyte is transformed into plasmocyte, while of the mice injected with interleukin 4. What do this result reveal? Because B lymphocyte of the mice injected with interleukin 4 and only the B lymphocyte of the mice injected with interleukin 4 are transformed into plasmocyte. So, interleukin 4 is indispensable for differentiation B lymphocyte into plasmocyte. So, out of the two interleukin secreted by active T4 lymphocyte, only interleukin 4 is indispensable for the differentiation of B lymphocyte into plasmocyte. To study the mechanism of the elimination of the antigen, SRBCS are added to the serum taken from mice of lot 1. The observation of the aspect suspension shows agglutination of the SRBC by the formation of immune complexes. These immune complexes are then spotted in 3 different cultures. Many experiments are performed where the condition and the result are showing in document 2. So, after the plasma cells produce antibody, the antibody bind to the antigen. As a result, an immune complex is formed. For culture 1, only the immune complex is present. The result, the immune complex, remain unchanged. For culture 2, macrophage are added to the immune complex. The result is the immune complex disappear because the macrophage eliminate by phagocytosis. For culture 3, complement protein are added to the immune complex. So for the result, the immune complex also disappear because the complement protein destroy it by lysis. So, for question 7, explain the result obtained in culture 2 and culture 3. This is the answer. The disappearance of the immune complex in culture 2 can be explained by opsonization. The phagocytosis of the immune complex by the macrophage is facilitated by the antibody that fix the antigen, and the disappearance of the immune complex in culture 3 can be explained by the lysis of the cellular antigen after the activation of the complement protein by the antibody of the immune complex. So, for conclusion, the immune complex can be eliminated in two ways: by macrophage through phagocytosis and by complement protein through lysis. So, for final conclusion for question 2, the presence of antigen on the body fixed on the macrophage present the antigen to T4 lymphocyte and activated the T4 lymphocyte secretion of interleukin 4. The interleukin 4 activate the B lymphocyte, and the B lymphocyte differentiation for plasmocyte, and plasmocyte produce the antibody, and the antibody fix the antigen and form an immune complex, and the immune complex elimination by macrophage by phagocytosis or complement protein lysis. And thank you, and see you in the next video.